this stuff on the glycolysis, on the metabolism, let me give you an idea of some questions. Now, many of you I had in 2401, and some of these questions are just like we had in 2401 on metabolism. And metabolism, like, for example, a question like this. Glycolysis is the metabolic pathway by which glucose is converted to lactic acid, acetyl coenzyme A, phosphoglyceraldehyde, pyruvic acid, NAD reduced. All right, glycolysis is the metabolic pathway by which glucose is converted to who? Pyruvic acid, isn't it? Remember that? Y'all haven't studied that. You vaguely remember that? It's kind of coming back. Glucose to pyruvic acid. Now, also is glycolysis aerobic or anaerobic? Anaerobic. All right, now listen to this one. And all of you know that in metabolism, the building up is anabolism. Y'all know that, don't you? And the breakdown is catabolism. Everybody understands that part. All right, now look at this one. Now, what is biological oxidation? It's the loss of protons and electrons, or hydrogens, 2 at a time. Hydrogens 2 at a time. Okay, in other words, that would be, you could say the loss of H plus and E minus 2 at a time, right? Hydrogens 2 at a time. H plus, what's H plus? That's a proton. What's E minus? That's an electron. Hydrogens and electrons 2 at a time. Okay, so everybody is aware of that. Okay, now listen to this one. I like this one. If a person eats too many carbohydrates and doesn't get enough exercise, glyceraldehyde 3-phosphate may be converted to glycerol through the process of glycogenesis, glyconeogenesis, lipogenesis, glycolysis, or oxidation. What would it be? Okay, somebody said lipogenesis. Glyceraldehyde can be converted to glycerol. That's a step in forming the triglyceride, the fats. Okay? All right, now watch this one. Now, the anaerobic respiration produces how many ATPs? I'm sorry, wait a minute. Yeah, anaerobic ATP. Anaerobic respiration, anaerobic, keyword, produces how many ATPs for each glucose molecule that's catabolized? Anaerobic. What's the net gain of ATP, in other words, in going from glucose to pyruvic acid? The net gain is what? 2. Remember? Anaerobically, 2. Okay. All right, so be sure you got that one. All right, now look at this one. Beta oxidation and deamination are processes associated with the metabolism of, fats and carbohydrates, vitamins and minerals, carbohydrates and proteins, micelles and chylomicrons, are fats and proteins. All right, look at this. Beta oxidation and deamination. Beta oxidation, that's fat, isn't it? Deamination, protein. Would it be fats and protein? Fats and protein, beta oxidation and deamination? You kind of follow? Kind of see? Okay. All right, very good. All right, now then, before fatty acids can be oxidized by means of the Krebs cycle. Remember the fatty acids? And if we're going, before the fatty acids can be oxidized by means of the Krebs cycle, what would they have to be converted to? Now listen to this right here. Fatty acids, acetyl coenzyme A, pyruvic acid, lactic acid, amino acid, or phosphoglyceric acid. All right, fatty acid, beta oxidation, when they have to be converted to this, acetyl, when they have to be converted, fatty acid, beta oxidation, and then combined with the CoA, before fatty acids can enter the Krebs cycle, they got to be converted to acetyl coenzyme A. Acetyl coenzyme A, the coenzyme A is the ticket for these things to get into the Krebs cycle. Okay, so kind of remember that a little bit. All right, kind of, sort of. All right, now, glycolysis reactions occur in which part of the living cell? Endoplasmic reticulum, Golgi bodies, lysosome, cytosol, or mitochondria? Glycolysis occurs where? Good job, cytosol. Okay? All right, that's okay. Now listen to that. I like this one. Between meals, glucose level of the glucose level of the blood is maintained by transamination, deamination, lipogenesis, glycogenolysis. That is the beginning of the breakdown of glycogen into glucose. That is glycogenolysis. Okay, so remember those terms right in through there. This, again, I haven't worked on your exam on this one. I do have a thing right here on this. I do have something like this. Lipids are stored primarily as triglycerides, synthesized by beta oxidation, broken down by oxidative deamination, only made up of LDLs and HDLs. That's false. Synthesized by beta oxidation, stored. The lipids are stored, everybody, primarily. They're stored primarily as triglycerides. They are stored primarily as, just remember this, lipids are stored primarily as triglycerides. Okay? They're not synthesized by beta oxidation. Beta oxidation actually carries the 2 carbon fragments to form acetyl coenzyme A. They're not broken down by oxidative deamination because lipids don't have amine groups. Only made up of HDLs and LDLs? No. In other words, lipids, no. Lipids are made up of a lot of things. Triglycerides, cholesterol, various other things. All right, now look at this. All right, this one right here. Okay, the essential step in the catabolism of an amino acid is the removal of an NH2 group, which is known as, what would that be? The removal of an NH2. Deamination, decarboxylation, transamination, hydrogenation, or beta oxidation. What is removal of NH2? Deamination. Good job. Deamination is the removal. Okay? Now, make sure you know the sequence in the Krebs cycle. The sequence of the acids. Do you remember them? Okay, let's see if I can repeat them for you. All right, you can start anywhere. But now remember, the 2-carbon acetyl group combines with oxaloacetic to form citric. Citric, isocitric. Isocitric, alpha-ketoglutarate. Alpha-ketoglutarate, succinyl coenzyme A. Succinyl coenzyme A, succinic. Succinic to fumaric. Fumaric to malic. Malic back to oxaloacetic. And then fill in the in-betweens there. Know where CO2, know where the beer men come in. Remember how we talked about that? Know where those guys, where they come in. Okay? Like between succinic and fumaric, Mr. FAD plays a role. Between isocitric and alpha-ketoglutarate, Mr. NAD. Between alpha-ketoglutarate and succinyl coenzyme A, Mr. NAD. Then you come on around the horn. I already mentioned succinic, fumaric. Fumaric to malic, nothing. Malic to oxaloacetic, Mr. NAD comes in. Alright, so that's the way where those things, be sure you kind of know where those come in. What are the iron-containing compounds, Nikki, in the electron transport? Do you remember the name of those? The iron-containing what? Good job. Cytochromes. Cytochromes are the iron-containing compounds in the electron transport. Alright, so let me move on. That's giving you an idea of what to look for. In fact, here's a question on the test. FAD is the oxidizing agent in the Krebs cycle between which 2 of the following acids of the Krebs cycle? I just told you. FAD, succinic and fumaric would be the answer. All right, and then and then right here, the electron carriers. I just asked this question. Somebody came up with the answer. The electron carriers that we emphasized in the electron transport chain are NAD, cytochromes, FAD, acetyl coenzyme, hexokinase. Cytochromes. Cytochromes. They're what? Do they contain magnesium in the molecule or iron? Iron. Okay. They're very similar in construction to hemoglobin. All right, everybody good with that? Everybody's good. All right, let's go. Okay. All right, let's get to Das Blut. You know what Das Blut is? The Germans say it like this. Shasta, they say Das Blut.